How's it going, folks? Can you guys hear me okay? Anybody there?
Yeah, I can hear.
Thank you. Appreciate it, Jamie Lee. All right, guys, so let's get started. Today we are discussing about the urinary system, and we are getting super close to the end now. So we only have, after this one, we only have one more chapter, and we're going to be done. And then we're going to have our fourth exam. And during the finals week, we're going to have our final. Does anybody have any questions about the final exam? No questions?
Does it seem like the regular exams?
No, it's not the same, actually. It is different format. For the final exam, we have the critical thinking questions on Lamaku. So if you go under the important resources, there's a file there called the critical thinking questions. And there are about 20 questions in there. Out of those 20, I'm going to pick 8 of them for the final. Okay, so the final is going to be 8 questions from the critical thinking file that you have on Lamaku. And each one of the questions is going to be worth 10 points. So the final is worth 80 points total. Any other questions, folks? All right. So let me share my screen with you, and we can go over the urinary system. Give me a second here. All right, guys. So we have a lot of information to cover today, and the urinary system can be a little complex. Okay. So I need you guys full attention here. And second here, wrong file. So I need you guys full attention and please ask questions. If something is not clear, as usual, just stop me and let me know. Okay? Now, what are we going to discuss in this chapter? In this chapter, we're going to start by taking a look at the different organs of the urinary system. We're also going to talk about the physiology of the urinary system or the function of the urinary system. Why do we have one? What does it do for us? We're going to spend a ton of time today discussing about what we call the nephron. And I need you guys to understand the nephron already as we discussed more about it, because we will definitely be on the exam. So what is the nephron? The nephron is what we call the functional unit of the urinary system. Okay? Most of the important functions of the urinary system are performed by the nephron. Within the nephron, excuse me, this is where blood is going to be filtered and the urine is going to be formed. So we're going to talk about the process of urine formation, which happens within the kidneys, within the nephrons. And then we're going to end this discussion talking about maintaining balance in our body, specifically maintaining pH balance, which is another function of the urinary system. So let's start from the basic here, from the big picture. What are the organs or the main organs of the urinary system? The urinary system includes 2 kidneys. They are bilateral. So we have the right kidney, we have the left kidneys. And inside of the kidneys, this is where blood is going to be filtered and urine is going to be formed. Now, as the urine, they are formed inside of the kidneys, this urine, it needs to get out of the kidneys. And the urine is going to get out of the kidneys through 2 tubes that we call the ureters. And they are also bilateral. So we have the right ureter taking urine out of the right kidney, and we have the left ureter taking the urine out of the left kidney. From the ureters, now this urine is going to go inside a container, for lack of a better word, that is going to store the urine. And that storage of urine is done by what we call the urinary bladder. And we're going to take a look later how the walls of the urinary bladder, we're going to find a smooth muscle called the trusor muscle. And this muscle, when it's time to empty the bladder, this muscle is going to contract, pushing the urine out through what we call the urethra. Guys, very important. Do not confuse ureters with urethra. It happens very often. We have two ureters. We only have one urethra. Okay? Now, what are the functions of the urinary system? Well, one of the functions of the urinary system is for the regulation of electrolytes in our blood. Now, we have talked about electrolytes before. We know that electrolytes are charged ions, right? So potassium, sodium, calcium, magnesium, these are all different electrolytes. And our urinary system, specifically the kidneys, will regulate the amount of electrolytes in our blood. Make sure that it is at the homeostatic level. Our urinary system is also very important to regulate our blood volume, because we need to maintain the proper amount, the proper volume of blood in our body. Because remember, there's a direct correlation between the volume, blood volume, and blood pressure. When the blood volume goes up, blood pressure goes up. When blood volume goes down, blood pressure goes down. Okay? Our urinary system is responsible for the regulation of glucose in our blood through a process called gluconeogenesis, which is the formation of glucose not from carbohydrates, but from fatty acids and amino acids, so fats and proteins. And our urinary system helps to regulate the pH of our blood. I don't know if you remember, but our blood pH is slightly alkaline. So it varies between 7.35 and 7.45 in the pH scale. Okay? And the urinary system, we're going to take a look at this later, probably on Thursday, helps to maintain the proper pH of our blood. The urinary system also helps to get rid of what we call nitrogenous waste, such as ammonia, urea, uric acid, which is all, full packed with nitrogen, hence the name nitrogenous waste. And the urinary system, specifically the kidneys, are involved with the production of a hormone called erythropoietin. We have discussed this hormone before. We know that erythropoietin is involved with red blood cell production. The kidneys also produce another hormone called renin that helps to regulate our blood volume and blood pressure. And our kidneys help to activate vitamin D. Okay, so the vitamin D that we consume, the vitamin D that is produced with the cholesterol in our body, needs to be activated. And when the vitamin D passes through our kidneys, the kidneys will turn them on, will activate those vitamins. Now, let's take a look inside of the kidneys. First of all, the kidneys, together with the pancreas, together with the beginning of the digestive of the small intestine called the duodenum. So both of them, all of them, kidneys, pancreas, and duodenum, they have what we call a retroperitoneal position. What does retroperitoneal means? Retroperitoneal means that they are found behind the parieto-peritoneal membrane. Okay, so we need to cut through that membrane in order to expose the surface of the pancreas, of the kidneys, of the duodenum. Our kidneys have a very peculiar shape to it. Right in the middle of the kidneys, there is this notch or this indentation right here, where we have the attachment of the ureter, where we have the attachment of the renal artery and the renal vein. This region is what we call the renal hilum. So once again, what is the renal hilum? The renal hilum is the indentation in the middle of our kidneys. Okay? When we open up a kidney and we look inside, we're going to see a rim of a structure called the cortex. So this is the renal cortex. And we're going to see a better view of it in the next slide. The entire inner section of the kidneys, this is what we call the renal medulla. So again, the entire section is the renal medulla. Now, the renal medulla is divided into little lobes. Each one of these lobes is what we call a renal pyramid, which are separated from one another by renal columns. So let's take a look at those structures again. So here we have an actual kidney from a human cadaver. So we can see this lighter region right here on the outer rim. This is the renal cortex. The entire inner section is called the renal medulla. The renal medulla is divided into lobes called the renal pyramids, and the lobes are separated by this tissue right here, and that is the renal column. Okay? Now, inside of the cortex and the medulla, right in this region that I'm circling right here, guys, this is where the nephrons are going to be found. Okay? And the nephrons, this is where urine formation is going to happen. Now, as the urine is formed inside of the medulla right here, this urine needs to come out of the kidneys. And it's going to come out of the kidneys through little channels inside of the kidneys. And those channels have small different parts to it. The first section of the channel is what we call the minor calyx. Everybody look up here for a second, please. So guys, at the base of each pyramid right here, where the urine is going to start flowing, this is what we call the minor calyx. Okay? Little channel right here. From the minor calyx, now the urine is going to flow into a bigger channel, a wider channel. This is what we call the major calyx. And then from the major calyx, now the urine is going to flow yet to a wider channel. And this is what we call the renal pelvis. And once again, it goes minor calyx, major calyx, renal pelvis. Now from the renal pelvis, the urine is going to flow into the ureter, and then from the ureter is going to go urinary bladder, and finally exit the body through the urethra. So this is the flow of urine through our body. Okay, it started all the way inside the channels of the kidneys, and then finally coming out through the urethra. Now, we understand that one of the main functions of the kidneys is for the filtration of our blood and formation of urine. Now, how does this blood get in and out of the kidneys? This is what we're going to be discussing in the next few slides. Okay, so let's see what brings oxygen and nutrients inside of the kidneys and what is going to remove the waste from the kidneys. So we have a renal artery, which is going to bring blood inside of the kidneys, and we have the renal vein, which is going to take blood out of the kidneys. Remember that the renal artery, the renal vein, and the ureter, they are attached to the kidney at the renal hilum. So this region right here is what we call the renal hilum. So let's take a look at the flow of blood in and out of the kidneys. Okay, and we're going to go over each one of those blood vessels, and I number each one of them here for you so you're able to identify them. So the blood is going to start right here with the renal arteries, number one. Then it's going to flow through what we call the segmental artery, number two. This segmental artery is going to pass in between the pyramids, in between the lobes, and that's going to become what we call the interlobar artery, number three. And now from the interlobar artery, the blood is going to go through this artery right here, right on top of the pyramids. These are what we call the arcuate arteries, number four. And then the arcuate arteries, they're going to radiate through the cortex. And now we're going to call them cortical radiate arteries, number five. Now, the next four blood vessels right here, folks, we cannot see them in this slide because they are very tiny, very small. These are the blood vessels that feed blood in and out of the nephrons. Okay, and we're going to discuss them separately. Now, from the cortical radiate artery, now the blood is going to flow into the afferent arteriole, and from the afferent arteriole is going to flow into the glomerular capillary, also called the glomerulus. And I want you guys to make a note of the glomerular capillary here, because the glomerular capillary, this is where blood filtration happens. Okay, this is where our blood is going to be filtered. So now once the blood is filtered, the blood is going to exit the glomerular capillary and is going to go through the afferent arteriole. And from the afferent arteriole, it's going to flow through a very small capillaries that is wrapped around what we call the renal tubules. And this capillary is called the peritubular capillary. And the peritubular capillary, they also have a very important function. This is where we're going to have the reabsorption and secretion of things back and forth between the capillary and the renal tubules. And again, we're going to discuss more about them today. After the peritubular capillary, now the blood is going to flow into the veins. And it's going to start in the cortical radiate vein, number 6 right here. It's going to go through the interlobar vein, the arcuate vein, then the interlobar vein, and finally it's going to exit through the renal vein. Guys, just to be clear here, you do not need to memorize the name of these blood vessels right here, okay, or these ones right here. But you do need to know the name of these 4 blood vessels right here, the ones that are feeding blood in and out of the nephrons. So you need to know the afferent arteriole, glomerular capillaries, afferent arteriole, and the peritubular capillaries. All right? Don't worry about the other ones. Any questions, folks?
You said those ones feed blood to the nephrons?
Yes. These 4 in the middle here. Any other questions? All right. So let's take a look at the nephrons then. And remember, the nephrons are the functional unit of our urinary system. We have about 1 million nephrons per kidneys. And we need every single one of those nephrons because nephrons do not grow back. When our nephrons are destroyed, they are gone. It's just like the alveoli in the lungs. When you lose the alveoli, they're gone. They're not going to grow back. Okay? We have 2 different types of nephrons. We have what we call the cortical nephron, which is just found within the cortex, the renal cortex. And we have the juxtamedullary nephron, which is spanned from the cortex into the medulla. Hence the name juxtamedullary. Now, most of the nephrons in the kidneys, they are cortical. About 85% are cortical nephron. And the cortical nephrons, they are the most important. They are the ones that do the everyday work of the kidneys. Okay, filtration of blood, formation of urine. Now, the juxtamedullary nephrons, they have a function of concentrating the urine. Okay, that is the main function of that nephron. So we're going to focus mostly on the cortical nephron. All right, but they all have the same parts. The only difference really is that the juxtamedullary nephrons, they have a longer, what we call loop of Henle. We're going to talk more about it in a minute. Now, the nephron is made of different parts. This circular region right there that I circled for you, this is what we call the renal corpuscle of the nephron. And within the renal corpuscle, this is where blood filtration and the urine formation is going to happen. Now, as the blood is filtered and the filtrates start to form, this filtrate needs to get out of the renal corpuscle. And it's going to get out of the renal corpuscle through a series of renal tubules. And there are 3 different sections, which we're going to talk about right now. So let's see what are the parts of a nephron. Okay? So again, a nephron is made of a renal corpuscle, which is this circular region right here. We're going to take a closer look at it in the next slides. And the renal corpuscle itself is made of 2 different structures. It is made of a blood vessel called the glomerular capillary, all the way on the inside, and is made of a capsule surrounding the glomerular capillary. And this capsule is what we call the glomerular capsule, also called Bowman's capsule. Now, as I mentioned in the previous slide, in the renal corpuscle, this is where blood filtration and urine formation is going to happen. And as we have the formation of this fluid called the filtrate needs to get out of the renal corpuscle. And it's going to get out through 3 different sections of renal tubules. The first section is what I call the PCT, and I color-coded them here for you in green. This is what we call the proximal convoluted tubule, all right, which is directly attached to the glomerular capsule. Then the filtrate is going to go through what we call the loop of Henle, and the loop of Henle has a descending limb and an ascending limb in which the filtrate is going down and up. And then from the ascending limb of the loop of Henle, now the filtrate is going to go through the DCT, or the distal convoluted tubule, shown here in blue. And guys, these are the 3 sections of the renal tubule: PCT, loop of Henle, and DCT. Okay, and this will be on the exam. So you need to know and be able to identify the different structures of the nephron. After the filtrate passes through the DCT, now the filtrate is going to go through what we call the collecting duct. Okay, and then at the end of the collecting duct, what we have, guys? We have the minor calyx. So the minor calyx is going to be right there, which is going to receive the urine. So now when the fluid gets in here, in the minor calyx, now we're going to call that fluid urine, because now is the final urine that we're going to expel out of the body. And remember, it goes minor calyx, major calyx, renal pelvis, ureter, and then urinary bladder, and finally urethra, okay? And then it comes out of the body. So what I want to do right now, and I need everybody's attention here so you understand the next slide. I'm going to take a little slice of the renal corpuscle and the PCT, the proximal convoluted tubule, okay? That's what we're looking at right here. So this is the renal corpuscle, and this little section right here is the PCT, the proximal convoluted tubule. Now, remember that our 4 blood vessels you guys need to know for the exam, right? Blood vessels that are feeding blood to the nephron. What are they, guys? We have the afferent arteriole, and again, the afferent arteriole takes blood inside of the renal corpuscle. Then the blood is going to pass through the glomerular capillary, aka glomerulus. Then the blood is going to exit through the afferent arteriole. And finally, the blood is going to pass through the peritubular capillary. Those are the blood vessels that you guys need to know for the exam. Okay? And so as the blood passes through the glomerular capillary right here, guys, this is where blood filtration is going to happen. And as the blood is filtered, the fluid from the filtration of the blood, we call it a filtrate. It's not the urine yet, because it's not the final composition of the urine. Okay, so we don't call it urine, we call it the filtrate. And this filtrate is going to start to accumulate right here in the space of the glomerular capsule. As the filtrates accumulate, now this filtrate is going to flow into the renal tubules. It's going to pass through what? It's going to pass through the PCT, the loop of Henle, and the DCT, the distal convoluted tubule. And then this filtrate is going to enter the collecting duct. Okay? Now, how do we filter our blood, guys? This is a very important question. This is a question that will be on the exam. I will need everybody's attention to this little figure right here at the bottom. Okay, we are looking at a cartoonish representation of the renal corpuscle. So we can see the afferent arteriole bringing blood in, and we can see the afferent arteriole taking blood out. Now here in the middle, we have the glomerular capillary. And we can see that covering the glomerular capillary, there is this yellow tissue right here. What is this yellow tissue? Guys, this yellow stuff on top of the glomerular capillary, these are cells. Cells that we call podocytes. Podo means foot. Cyte means cells. So these little cells, they have foot processes that actually look more like fingers. Okay? And these foot processes are called the pedicels. And those foot processes, when they interlace with one another, just like what I'm doing with my hands right here, they are going to create little gaps right here. And these little gaps are called the filtration slits. Okay? Now, anything that is small enough to pass through the filtration slits is going to be part of the filtrate. Anything that is too big to pass through the filtration slits will stay within the blood. I want you guys to think of the podocytes and the filtration slits like a coffee filter. I don't know if any of you ever made coffee at home, right? In which you have a paper filter, you put the coffee grind on the filter, you take boiling water and you pour over it. What do you have on the other end? You have coffee, which is the filtrate. So this is what is going to filter our blood. It's going to be the podocytes and the filtration slits. Any questions, guys? All right. Now, here we have again another look at the renal corpuscle, a much better view of the renal corpuscle, both in the external view as well as an internal view. So we can see again the afferent arteriole, and we can see the afferent arteriole. How can we tell them apart, guys? What if this was not labeled here? How can we say which one is which? Well, let me tell you. The afferent arteriole is wider. It is a wider blood vessel than the afferent arteriole. The afferent arteriole is thinner, okay? The afferent is thicker. Now, again, as the blood goes into the afferent arteriole, the blood is going to enter the glomerular capillary. The glomerular capillary are covered with podocytes, which creates the little filtration slits. Whatever is small enough to pass through the filtration slit is going to become part of the filtrate. And this filtrate is going to flow through the renal tubules, including the proximal convoluted tubule. Now, right here, we can also see two important cells that I want you guys to know for the exam. And those cells are the juxtaglomerular cells, which are found in the afferent arteriole, and the macula densa, which we find in the DCT, the distal convoluted tubule. These cells right here, they have a very important function with our blood pressure and our blood volume. So they help to regulate blood pressure and regulate blood volume. Okay, we're going to talk more about them in a minute. Now, putting the whole thing together, putting the renal tubules together with the blood supply, this is what they would look like. So we have afferent arteriole, and then the peritubular capillary wrapped around the renal tubules. Okay? And I want to talk about this region right here that I circled for you in blue. This is what we call the juxtaglomerular apparatus, or the JGA. And the JGA includes those two cells that we just talked about in the previous slide. We have the macula densa, and we have the juxtaglomerular cells. The macula densa cells, guys, they are responsible for sensing the levels of salt, sodium chloride in our blood. While the juxtaglomerular cells, they are responsible for sensing our blood pressure. Okay? So one senses the level of salt, the other senses the level of the pressure. Now, what are they going to do? How are they going to control our blood pressure? They're going to control our blood pressure by controlling the blood volume. Because again, remember, there's a direct correlation between the blood volume and the blood pressure. If blood volume goes up, the blood pressure goes up. Blood volume goes down, blood pressure goes down. Okay? Now, once again, the nephrons cannot grow back. Once they are damaged for life. And eventually, if a lot of your nephrons start to die, guys, what's going to happen to your kidney function? Your kidney function is going to stop. And you're going to have to be put into a dialysis machine in order for you to be able to perform the functions of the kidneys. So let's take a look now at how urine is formed inside of the nephrons, okay? And I want you guys to write this down because this is going to be on the exam. Urine formation involves three different steps, three different processes. Urine formation involves what we call glomerular filtration, tubular reabsorption, and tubular secretion. So these are the 3 steps of urine formation. Glomerular filtration, which is going to happen right here in the glomerulus, in the glomerular capillary. This is where the blood is going to be filtered, and the filtrate is going to be formed. Then, as the filtrate passes through the renal tubules, we're going to have a tubular reabsorption and tubular secretion. What does tubular reabsorption means? Guys, tubular reabsorption, it means that there is stuff in the filtrate that got filtered out, like glucose, for example. Glucose is small enough to pass through the filtration slits, but we want the glucose back into our blood. We need the glucose. We want the glucose. Another example, amino acids. Amino acids are small enough to pass through the filtration slit, so they're going to become part of the filtrate, but we want them back into the blood. We don't want to get rid of them. This is going to happen through what we call tubular reabsorption. Now, what about tubular secretion? Tubular secretion is when we have stuff in the blood that we don't want in there. So we're going to send it into the renal tubules so that it can become part of the filtrate and we can dispose of it through the urine. Okay? So let's take a closer look at the glomerular filtration. And again, glomerular filtration is done by the podocytes that are covering the glomerular capillary, and they are going to form the little filtration slits. We can see those little slits right here. Now, this filtration is going to form the filtrate, which is whatever is filtered from the blood. Okay? And the filtrate is going to be pushed out of the glomerular capillary due to what we call hydrostatic pressure, which is the water pressure inside of the glomerular capillary. Now, can blood cells and proteins pass through the filtration slits, guys? The answer is no. Blood cells and proteins are too big to pass through the filtration slits. This is why usually we don't find the proteins in our urine. We don't find the blood in our urine because the blood cells cannot pass through the filtration slits. If we find the blood in our urine, it's because there is something wrong down the line in the urinary tract. Okay? Now let's take a look at the tubular reabsorption. What is going to be reabsorbed back into the blood from the filtrate into the blood? Glucose, amino acids, ions, and bicarbonate is going to be reabsorbed back into the blood. We want them. What is not going to be reabsorbed? Urea, uric acid, creatinine. These are all waste products that we don't want in the blood. Okay? Now, what about tubular secretion? What is going to be secreted? Hydrogen ions are going to be secreted, guys. Excess hydrogen ions that are in the blood will be secreted into the renal tubule, into the filtrate. And this is going to help us to control, to regulate our blood pH. Remember, the blood pH needs to be slightly alkaline. Too much hydrogen ions cause our blood to become acidic, okay? So the blood varies between 7.35 and 7.45 in the pH scale, slightly alkaline. Also, we're going to secrete potassium, and we're going to secrete ammonium and ammonia, as well as creatinine and toxins and drugs. So these are all stuff that we don't want in the blood, we're going to send them into the filtrate so that we can dispose of it. Okay? Any questions, guys? All right, so let's take a look at this video here, which is going to give us an overall view and summarize of the function and the parts of the urinary system.
My feet are killing me. But they don't have to. Dr. Scholl's new life for your feet.
Planet Fitness. We're all strong on this planet.
One more, everybody.
We've been spending a lot of time lately talking about eating and digesting and metabolizing food. And those are some of my favorite things in the world. It's been a really great time. But as with all good parties or brunch buffets, in the end, we're left with a mess. And I'm not talking about spilled beer and Dorito crumbs. I'm talking about toxic levels of garbage that need to be cleaned up before they kill you. In your body, a lot of the cleanup that comes after metabolism is handled by the liver, which plays a tremendous role in directing dead cells and leftover chemicals to the digestive and urinary systems. But your liver can't actually escort waste out of your person. Your lungs can lend a hand exhaling carbon dioxide, and of course your colon will eventually poop out unusable stuff and old cell parts. But much of your chemical waste still needs to be sorted and disposed of, so one system steps in to bat cleanup. And that is your urinary system. This system, and specifically your kidneys, does all sorts of important homeostatic stuff like regulating your water volume, ion salt concentrations, and pH levels, and influencing your red blood cell production and blood pressure. But its main purpose, what we're going to be focusing on for the next 2 lessons, is how it filters toxic leftovers from your blood, like nitrogenous waste made by metabolizing protein, and ferries it out of the body. And spoiler alert, this all involves the how and the why and the what of your pee. Now, you probably know that kidneys are filters, and you may imagine them as sieves that strain out the bad stuff, leaving it sitting like a hair above the bathtub. But that's in fact kind of the opposite of what you should be thinking. Most of what's in your blood is totally removed by the kidneys, then your body pulls what it wants to hold on to before the rest is sent on a one-way trip to the bladder. It's kind of like this. You don't clean out your fridge by just taking out the rotten, fuzzy leftovers. Instead, you gotta take everything out and put it on the counter and then sort what goes back into the fridge and what goes in the trash. That's how your urinary system cleans you up, and it is really good at its job. So this morning, I decided to go the healthy route, and instead of eating my normal breakfast of nothing, I had a big 32-ounce protein smoothie. My digestive system did its thing, and all the protein was hydrolyzed into amino acids, which were absorbed by my body to build and repair cells. It's a beautiful thing, but not without consequence, because metabolizing nutrients, especially protein, makes a mess. Amino acids are unique in that they have nitrogen in their amino groups, and because they can't store amino acids, extra ones get processed into storable carbs or fats. The amine group isn't used in those storage molecules, so it's converted to NH3, or ammonia, which happens to be toxic. So the liver converts the ammonia into a less toxic compound, urea, which our kidneys filter out into our pee. Once out of the body, urea can degrade back into ammonia, which is why dirty pee-soaked toilets and cat litter boxes smell like ammonia. Now, this business of taking out the nitrogenous trash is one of the urinary system's biggest jobs. Its other major duty is to regulate the balance of salt and water in your blood, and both of these tasks are processed in the whole system of tubes that is your urinary system. So let's take a look at some basic pee-making anatomy. Your kidneys are a pair of dark red, fist-sized, bean-shaped organs that sit on each side of your spine against the posterior body wall. Kidneys are retrocadial, which means they lie between the dorsal wall and the peritoneum, a membrane that surrounds the abdominal cavity, rather than inside the cavity itself, like your intestines and stomach do. Each kidney has 3 distinct layers, beginning with the outermost core. Beneath that is the medulla, a set of cone-shaped mass tissue that secrete urine in tiny sac-like tubules. And finally, the innermost layer is the renal pelvis, a funnel-shaped tube surrounded by spindle that uses peristalsis to move urine out of the kidney into the ureter and into the bladder. Because the kidney's main job is to continuously filter blood, they end up seeing a lot of it. In fact, at any given moment, they hold over 20% of your total blood volume. Oxygenated blood enters the kidney through the large renal arteries, which deliver nearly a quarter of all blood pumped through the heart every minute. That means your kidneys filter about 120 to 140 liters of blood every day. The kidneys branch many times, ending in tons of little capillary groups. So a kidney isn't just one big filter. Instead, each one is made up of about a million microscopic filtering units called nephrons. Structurally and functionally, nephrons are where the real business of blood processing, which like pee making, begins. In 3 steps: filtration, reabsorption, and secretion. Each nephron consists consists of a round renal corpuscle resides up in the cortex, followed by a long and winding renal tubule that loops around between the cortex and the medulla. The outer part of the corpuscle is a cup-shaped feature called the glomerular capsule, because inside it there's a whole angle of capillaries called the glomerulus. That's from the Latin word for ball of yarn, which is pretty looks. And the endothelium of these capillaries is very porous, so they allow lots of fluid, waste products, ions, glucose, and amino acids to pass from the blood into the capsule. But they out bigger molecules like blood cells and proteins, so they stay in the blood and exit through the peritubular capillaries, also known as the vasa recta. Now, all the stuff that gets squeezed out of the blood into the glomerulus is called filtrate, which is then sent along the elaborately twisting 3-centimeter long renal tubule. Even though it looks like just a tube, it actually has 3 major parts, some of which are permeable to certain substances but not others. First is the proximal convoluted tubule, or PCT, which is about as convoluted looking as its name suggests. Then the tube drops into a dramatic hairpin turn called the nephron loop, or the loop of Henle. And finally it ends in the distal convoluted tubule, or DCT, which empties into a collecting duct. All this twisting might make the tubule look like super inefficient, but it actually serves an important purpose, as you might expect. Just like with your small intestines, the long, curly shape of the nephron provides more time and space for it to reabsorb whatever usable stuff it can. And this meandering path allows the parts of the tubule that are toward the end to have an effect on processes that take place closer to the beginning as they pass each other. Because a lot of the stuff that winds up in the tube are valuable commodities, like ions and glucose and water, and we don't want to just pee all of them out if we can help it. So let's trace the whole process, starting at the top with the proximal convoluted tubule, or PCT. The walls here are made of cuboidal epithelial cells with big ol' mitochondria that make ATP to power pumps that pull lots of sodium ions from the filtrate using active transport. These cells also are covered in microvilli that increase their surface area and help reabsorb much of the good stuff from the filtrate back into the blood. The remaining filtrate passes from the PCT into the loop of Henle, which starts in the cortex, then dips into the medulla before coming back into the cortex. And the form of this loop is key to its function, because its primary task is to drive the reabsorption of water by creating a salt concentration gradient in the tissue of the medulla. It does this mainly by actively pumping out salts in the ascending limb. This creates some very salty interstitial fluid in the medulla, so when new filtrate comes down the descending loop in front of it, water passively flows out and into the super salty interstitial space. Since most of this water is picked up by the blood pretty quickly, the saltiness of the interstitial space doesn't get diluted, so it can keep drawing water out of the next batch of filtrate in the descending limb. Needless to say, this is super important, because if we peed out all the water that went into our kidneys, we would die of dehydration really quick. But even after all that, we are still only 2/3 of the way through the process. As we move out of the loop of Henle into the distal convoluted tubule and onto the collecting duct, the remaining filtrate is now officially urine. But there's one more component that we have to squeeze the most out of before we excrete the stuff: urea. Even though we think of urea as a waste product, just one more part of that protein shake that has to be dumped, the kidneys actually need it. They use it to raise the concentration earlier in the process, making the medulla even saltier for the filtrate that's back there going through the ascending limbs. When the final step, after the filtrate leaves the DCT, it enters the collecting duct, which runs back into the medulla. And while the salt passively leaves more water out of the collecting duct, some urea passively leaves the urine as well, making the medulla even more salty, in turn more effective at drawing out water from the ascending limb a few steps back. So there's essentially a traveling pool of urea that escapes the urine, finds its way back into the loop of Henle, and then runs the whole course again back to the collecting duct. An ammonia-scented cycle called urea recycling. Now all that's left is a kind of last call to selectively sneak out any extra waste like hydrogen, potassium, and certain organic acids and bases using active transport. This is called tubular secretion, and it transports only select kinds of waste that have already made their way into the blood that's in the peritubular capillaries ready to leave the kidneys. This step is kind of like emptying your pockets of any last wads of tissue or crumpled receipts as you're walking a bag of trash to the curb. And that's how your kidneys clean up the mess left over from the giant party that is you metabolizing food. So if you thought that your kidneys were just a kind of fine mesh that filtered out bad stuff, now you know that that's not true. If you thought your urinary system was basically a matter of water goes in, pee goes out, that's definitely not true. And if you thought we were done talking about your urine, that is also not true, because next time we're going to be learning how your body regulates what's absorbed and what's excreted, and we'll find out what can happen when that regulation goes awry. But for now, you learned the anatomy of your urinary system and how your kidneys filter metabolic waste and balance salt and water concentrations in the blood. Specifically, you learned how nephrons use glomerular filtration, tubular reabsorption, and tubular secretion to reabsorb water and nutrients back into the blood and make urine with the leftovers. Thank you to our headmaster of learning, Linnea Boyev, and thank you to
All right, guys. Any questions, folks?
all of our Patreon patrons.
All right. So the next section that we're going to talk about in this chapter, we're going to be talking about the composition of urine. We already understand how urine is made. Just to recap, urine formation involves three steps, right? We have glomerular filtration, we have a tubular reabsorption, and we have a tubular secretion. And through those processes, we're going to have the formation of urine. Now, what is the composition of urine? What are we going to find within our urine? Normal urine, folks, output is about between 1 to 2 liters of urine a day. So this is how much we pee in a day, how much water we lose through our urine. Now, if it wasn't for the ADH, the antidiuretic hormone, we would pee a total or pass a total of 25 liters of urine a day. I don't know if you guys remember the ADH. We have talked about it before when we discussed the endocrine system. But the ADH stands for antidiuretic hormone, and it is a hormone that is produced by the hypothalamus and released by the posterior pituitary. And this hormone helps with controlling the amount of fluid that we lose through our urine. Okay, so if for some reason your hypothalamus or your posterior pituitary was not releasing ADH, you would become constantly dehydrated because you would pee about 25 liters a day. Our urine is light yellow in coloration, and that is because of the presence of this pigment called urochrome from bile and urobilin from hemoglobin. And we all know that when we haven't drank enough water in a day, our urine is going to be dark yellow. So it becomes very concentrated with the solutes in the urine. You're going to be surprised to know that fresh urine does not have a strong bad smell. It's only when urine sits and then bacteria colonizes it and it starts to break down the nitrogenous waste from the urine, then it's going to give off the very strong ammonia smell. Okay? Now, the reason for that is because actually urine is sterile. There should be no bacteria growing in our urine. The pH of the urine is much wider in variation in the range than our blood. Remember that our blood pH is between 7.35 and 7.45, while the pH of urine can vary all the way from 4.6 to 8, okay? And that is okay. You know, it depends on what we eat. It depends on what we drink throughout the day. And finally, the specific gravity of urine is greater than the specific gravity of water. What is specific gravity? Specific gravity is the amount of solutes dissolved in a fluid. We have a lot more solutes dissolved in urine than we have in distilled water. Okay. Now, what is a urine analysis and what are we going to find in it? I don't know if any of you guys ever done a urine analysis. What is a urine analysis? Urine analysis is when the doctor is going to ask you to check the components of your urine. And by looking at that, they can then tell the health of your kidneys, how well your kidneys are working, how well your kidneys are functioning. Okay? And usually, when you do a urine analysis, you have to be fasting at least 12 hours. So let's say you're going to do a urine analysis tomorrow. So tonight at 8 p.m., you stop eating, and then the next morning at 8 a.m., you go to the hospital or the clinic, you're going to pee in the cup, and then they're going to analyze your urine. Now, what are we going to find? What should we find in the urine, folks? Just to take a look here, we have about 180 liters of water passing through the glomerular capsule in a single day, a 24-hour period. 180 liters of water. Most of the water will be reabsorbed. It's going to be taken back into the blood. Otherwise, we would die. Our blood pressure and blood volume would be so low, we would die. we're only going to pass about 1 to 2 liters a day excreted through the urine. We have about 2 grams of proteins passing through the glomerular capsule. Most of it will again be reabsorbed. Very little is going to be excreted, especially because proteins are too big to pass through the filtration slits. Let's take a look at bicarbonate and glucose. What is bicarbonate? Bicarbonate is a buffer. We can see here 275 grams passing through the kidneys in a day. Vast majority will be reabsorbed because bicarbonate is going to help to maintain our blood pH. Glucose, 162 grams passing through the kidneys. All of the glucose will be reabsorbed. Guys, I have a question for you. If you do a urine analysis and you are fasting for 12 hours and you find high concentration of glucose in your urine, let's say 100 grams of glucose, what could that indicate? What disease could that indicate, guys?
Diabetes.
Diabetes, absolutely. Oftentimes, most people that are diagnosed with diabetes, they are diagnosed first by doing a urine analysis. Because even though our body wants all of the glucose back, and you can see here in a normal individual, we will take all of the glucose back. But when you are diabetic, you have a very high concentration of glucose in your blood. I don't know if you guys remember, but it can be as high as about 600 milligrams of glucose. And that is a huge amount, guys. Huge amount. And the kidneys simply can't take all of their glucose back. So a lot of the glucose will spill into the urine. This is why when you do a urine analysis and you are diabetic, even though you're fasting, you're going to see high levels of glucose in the urine because you have high levels of glucose in the blood. Okay? Now, what we should not find in our urine. We should not find glucose. If you do find glucose in the urine, this is a condition called glycosuria, and that can be due to diabetes, or a non-pathological reason is because you are eating too much sugar or drinking too much sugary drinks. We should not find proteins in the urine. Why not? Proteins are too big to pass through the filtration slits. If you do find proteins, this is a condition called proteinuria, and the pathological reason is because you have a inflammation of your nephrons, a condition called glomerulonephritis. Also, hypertension can cause proteinuria. Now, a non-pathological reason includes physical exertion. Let's say you finish running a marathon, you could find some protein in your urine, or pregnancy can also lead to proteinuria. You should not find pus in our urine, guys. What is pus? Pus is a combination of white blood cells and bacteria. If you do find pus in your urine, this is a condition called pyuria, and is a very good indication of a UTI, urinary tract infection. You should not find red blood cells in our urine. Why not? Blood cells are too big to pass through the filtration slits. If you do find red blood cell, it's a condition called hematuria, and this could be due to a bleeding in the urinary tract, due to trauma, kidney stone, or infection. You should not find hemoglobin in your urine. This, if you do find, there could be a transfusion reaction or hemolytic anemia. And you should not find bile pigment in our urine. If you do find bile pigment, that is a condition called bilirubinuria, and that is a very good indication of hepatitis, of liver disease. Okay? So by looking at our urine, we can also help with the diagnosis of different conditions, including conditions related with the liver. Now, as I said earlier, we do not regrow our nephrons. And if you are losing your nephrons, guys, how can we lose a nephron? Look, I don't know if you notice, but the little nephrons are very fragile. Remember, the little glomerular capillary is a very thin capillary inside the nephron. One of the main causes of damage to the nephron and to the glomerular capillary is hypertension and diabetes. Hypertension, because the blood is coming inside the glomerular capillary with such pressure that it can literally burst the glomerular capillary. Diabetes, because our blood contains so much solute, so much glucose, and those glucose can damage all the little structures of the nephron. As you lose nephrons, eventually your kidney is going to malfunction. It's not going to be able to perform its function anymore. At that point, you have two options only. You either get a kidney transplant or you're going to have to be placed on dialysis. What is dialysis? Dialysis literally means to separate. You're going to be hooked up to a dialysis machine, just like this one right here, in which they're going to be taking your blood and passing your blood through this machine right here. This machine is going to separate the waste from your blood and input back the glucose, the amino acid, the bicarbonate, etc. It's going to perform the function of your kidneys. Okay? And guys, when you're doing dialysis, this is not fun and games. If you have to be on dialysis, you're going to have to go to the dialysis center about 3 times a week, between 4 to 8 hours every time you go. It's like having a part-time job. Okay? And, I mentioned this before, we have such an epidemic of diabetes, type 2 diabetes in Hawaii for a while now. And I remember, I live in town in Honolulu, and I remember driving through places where used to be, I don't know, gas station or a veterinarian clinics, and they all got converted into dialysis centers because we don't have enough dialysis center to service all the people in the state, all the population in Hawaii that is developing type 2 diabetes, and they need dialysis. Okay, it is a serious situation, folks. All right, I'm going to stop right here. We are a good way through this. I know we covered a lot of information today. Make sure that you guys are taking a look at those PowerPoint slides before you come to class and again after, so that the information is retained in your brain, okay? On Thursday, we're going to finish this chapter, and we're already going to start our final chapter, which is chapter 16, the reproductive system. And then on Thursday, I'm going to open the quiz for chapter 15, okay? Have a great rest of your day, folks, and I'll see you then.
Thank you.
Thank you.